there's essentially a question of to what degree genetics impacts whether or not an individual ends up lean or overweight or obese or wherever they fall on the spectrum. And this is an interesting one because I think anecdotally, most people can say that they've seen, for whatever reason, some individuals just seem to be able to either lose weight or gain weight or maintain their weight better or worse than other individuals. So this paper we're going to look at today is an interesting perspective on this topic for a few reasons. First off is that it looks at individuals who are identical twins. So they have the same exact genetics, and it compares outcomes within pairs of twins to outcomes between individuals in the study that aren't genetically related. So we can see how genetics impacted the outcomes in the study. And the other interesting piece, or one more interesting piece about this study, is that it was extremely well controlled. And at least in my experience, you don't really see studies like this happening in more recent years. This one took place back in 1994, and they took these 7 pairs of identical twins and they observed them 24 hours per day for about 117 days. First, interestingly, we see that there's significant variance between individuals in terms of weight loss. So if we return to our questions at the beginning of this video, we see that different individuals will lose different amounts of weight, and that fat mass loss will vary in terms of distribution around the body. And I guess part 2 of my takeaways is that it appears genetics, at least based on this data, does significantly influence those outcomes, those differences. The additional interesting aspect of this is that although genetics does play seemingly a large role based on this data, it doesn't play a complete role in that even though there were differences between different pairs of identical twins, there were also differences within pairs of identical twins. Hey everybody, welcome to another episode of the Talking Studies series where I break down individual interesting research papers that I've read. Today we're going to take a look at a paper called The Response to Exercise with Constant Energy Intake in Identical Twins by Bouchard and Colleagues from 1994. Before we get into that, it's important to note that we shouldn't over-extrapolate or overemphasize any individual paper, and that it's important to take a look at an overarching body of evidence when it comes to looking at any given topic. With that said, first, a little background on this paper. I came across it during a kind of ongoing deep dive I have into research on weight loss and how to minimize fat-free mass or lean body mass losses during weight loss, those metrics being important because they're proxies for muscle mass. And during ideal weight loss for most people, if not all people, you'd be losing mostly, if not all, fat mass and minimizing the losses of muscle mass in the process. And the reason I found this paper interesting, or maybe a little background before I get into the paper itself, is that in recent years, I've heard the topic of how genetics influences weight loss or weight gain or weight management, or whether or not individuals become overweight or obese more and more often. And so there's essentially a question of to what degree genetics impacts whether or not an individual ends up lean or overweight or obese or wherever they fall on the spectrum. And this is an interesting one because I think anecdotally, most people can say that they've seen, for whatever reason, some individuals just seem to be able to either lose weight or gain weight or maintain their weight better or worse than other individuals. And so the question arises is to what degree this is reflecting genetics versus to what degree it's reflecting individual choices and whether or not some people just have better willpower than others or something like that. And when looking at this, these differences or how genetics might influence weight management, weight gain, weight loss, there are kind of 2 perspectives or 2 main perspectives I've heard. One is that genetically, some people might be predisposed to have higher cravings or higher amounts of hunger that would make it either more or less difficult for them to gain or lose weight. The other piece of the puzzle here that this study will look at today focuses more on is the degree to which genetics might influence an individual's energy expenditure, in particular their resting metabolic rate and the energy they expend during or via NEAT, which is non-exercise activity thermogenesis, which is essentially unintentional movements throughout the day. So some people might twiddle their thumbs, for example, or tap their feet, or kind of fiddle around. And those small movements throughout the day are burning energy, and some individuals may carry out these movements more or less than others. And so this paper we're going to look at today is an interesting perspective on this topic for a few reasons. First off is that it looks at individuals who are identical twins. So they have the same exact genetics, and it compares outcomes within pairs of twins to outcomes between individuals in the study that aren't genetically related. So we can see how genetics impacted the outcomes in this study. And the other interesting piece, or one more interesting piece about this study, is that it was extremely well controlled. And at least in my experience, you don't really see studies like this happening in more recent years. This one took place back in 1994, and they took these 7 pairs of identical twins and they observed them 24 hours per day for about 117 days at a research laboratory or kind of location where they had them living. And the reason this is interesting is because if you look at most papers, especially in more recent years, looking at kind of weight loss or diet or lifestyle interventions, they are either extremely short, so they'll just have like individuals staying at a lab for a week maybe, or they are less controlled in that they're free living. So, for example, some of the papers I read looking at artificial sweeteners and weight loss is they would give subjects either drinks or foods that contain artificial sweeteners or don't, and then they would just instruct them to consume them or not consume them, whatever the protocol was, at their home. So they weren't monitoring them 24 hours per day like the individuals in this study we're talking about today. Anyways, that's one other really interesting piece about this study. And the last one is that this study looked at weight loss induced entirely via exercise. So, since they were monitoring these subjects 24 hours a day, they were able to essentially identify the amount of calories they would need to consume just to maintain their weight without any exercise at all. And then, based on that, and based on how much energy they were expending via exercise, they were able to test whether or not they could successfully induce weight loss via only exercise alone in terms of creating the caloric deficit. Um, a little quote from their intro, quote, "Regular exercise is an important component of a weight control program and may be useful in the treatment of obesity. However, it has obvious limitations as shown by several studies carried out on voluntary free living subjects. To assess the specific influence of regular exercise on body mass, body composition, regional fat distribution, and relevant metabolic characteristics, one should ideally keep energy and macronutrient intake constant for the duration of the exercise protocol. This requirement is quite difficult to meet in free living individuals, and as a result, very few long term studies have been reported on this topic. In an earlier experiment, we had attempted to apply such controls in a short term negative energy balance protocol with 6 pairs of identical twins. We have described elsewhere the response of 5 young men to a 100-day exercise training protocol on constant energy intake. We have used the same general protocol in a new study designed to investigate the individual differences in response to negative energy balance caused by exercise and also to assess the importance of genetic similarity versus genetic heterogeneity in the response variability, end quote. So interestingly, this lab group performed a couple of other studies similar to this one, as they mentioned one very similar to this one, where they seemingly used pretty much the same protocol looking at exercise in individuals for weight loss, and another one where they almost did the inverse, where they identified the maintenance caloric intake for a group of individuals who I believe were identical twins as well, and they actually overfed them 1,000 calories a day or something like that, and looked at the differences within and between twins in the study based on weight gain. And that paper is called "The Response to Long-Term Overfeeding in Identical Twins." And that one I did read, which is also very interesting. Maybe I'll cover it in a future episode. Anyways, for this study, looking at some of the methods and kind of the design, like I described, they took these twins, they observed them for 24 hours a day. First, there was a run-in period where they had the subjects eat ad libitum, so eat whatever and how much they want, um, provided the researchers monitored everything they were eating so they could track every calorie, pretty much all the calories they were consuming. And then they measured their weight and their body composition over about 2 weeks during this period. And through this process, they were able to see, based on how much the subjects were eating, whether or not their weight was changing, whether it was going up, down, or staying the same. And they could create a fairly accurate estimate of the baseline caloric intake these subjects would need without any exercise just to maintain their weight. And they performed, a handful of tests during this period for baseline. And then for 93 days, roughly, I believe, they had the subjects exercising on a cycle ergometer, which, as I understand it, is pretty similar to just a stationary bike. And they were cycling twice per day, an hour each. So an hour in the morning and an hour in the afternoon or evening. It wasn't exactly an hour. It was however much time it took them to expend 1,000 calories total throughout the day. So like 500 in each session. And that was based on, I think, VO2 max tests to see how much energy they were burning while using the cycle ergometer at different resistances. And so it wasn't exactly an hour, but it came out to about an hour in each session across the study period. Few quotes here. Quote, 3 to 4 pairs of twins were studied at a time, and the study was completed in about 2 years. Each pair had to be available 24 hours a day for a period of approximately 4 months, a mean of 117 days. They were then housed as inpatients in an experimental forestry research station located in a provincial park, a wildlife reserve area about 80 kilometers from the Laval University campus. They were under 24-hour supervision by members of the project staff living with them. On the average, each man stayed in the unit for 117 consecutive days. 17 days for the assessment of the energy cost of weight maintenance and baseline testing, 93 days for the negative energy balance period, and 7 days for testing after the experimental protocol. End quote. Moving on a little further, quote, for the duration of the experiment, the daily food intake was individually controlled by nutritionists to make sure that each subject ate only the amount of energy prescribed. The food plate of each meal for each subject was prepared by the nutritionist and eaten in the presence of at least 2 members of the research personnel to ensure full compliance. End quote. So as you can see, the food intake aspect of this study was extremely well controlled. The nutritionists were preparing their plates, and there were at least 2 personnel watching them eat all of the food and tracking everything they were consuming at each meal. Moving on a little further, now looking at the exercise training protocol, quote, the daily energy deficit was induced entirely by exercise training performed twice a day on a cycle ergometer, one session in mid-morning and one in mid-afternoon, over a period of 93 days. On the average, subjects performed 160 exercise sessions over the 93-day period. Exercise sessions lasted 57 minutes, and exercise intensity ranged from 50 to 55% of VO2 max. Intensity was individually determined and carefully monitored using heart rate at all times during each session. The prescribed power output required to meet the criterion of 4.2 megajoules per day or 1,000 calories per day. Exercise energy expenditure above resting energy expenditure was derived from the oxygen uptake power output relationship obtained during a pre-experiment exercise tolerance test. The relationship was verified every 25 days and the exercise prescription adjusted accordingly. The exercise program was conducted in cycles of approximately 9 consecutive days of exercise, followed by a day of rest. There were 9 days of rest throughout the protocol, end quote. So just like that quote described, they were on the cycle ergometer twice per day for about an hour each time, cycling at 50 to 55% VO2 max, which is a pretty low, just aerobic pace. And every 25 days, they completed an exercise tolerance test, which, as I understand it, is pretty much similar, the same thing as a VO2 max test, where they gradually increase the tempo or the speed or the resistance on whatever device they're using to test the VO2 max. And they'll check how much oxygen is being taken in and how much CO2 the subject is giving out, as well as they'll track their heart rate and things like that to look at the peak amount of oxygen the subjects can consume, or rather can utilize, which is a measure of cardiorespiratory fitness. And they based the power output and pretty much the resistance on the cycle ergometer off of those measurements. Now, the last notable thing, I think, in the methods and design is the statistical aspect of the paper, in particular, the intraclass correlation coefficients and the F ratios. So I'll start off with just a quote and let them explain it, since I'm not a statistical whiz and will probably make you more confused than when we started if I just go off trying to explain this on my own. Quote, the intraclass correlation coefficient for the changes brought about by negative energy balance was computed from the between pairs and within pairs means of squares. This coefficient provides a quantitative estimate of the similarity within pairs in the response to the protocol. An intraclass correlation coefficient close to 1 indicates a perfect within-pair resemblance in response, whereas a coefficient close to 0 would imply that there was no within-pair resemblance in response to the treatment. Similarly, a high F ratio indicates a high ratio of the variance between pairs to the variance within pairs in response to negative energy balance, whereas A low F ratio, close to 1, indicates that the variances in response between and within pairs of twins are comparable, end quote. So one important thing here is describing that when they refer to within pairs, they're referring to a comparison between identical or within identical twins. When they're talking about between pairs, they're looking at comparisons between individuals that are not genetically related. So between, if you take one individual twin, for example, a within pair comparison would be comparing that twin to their identical twin. A between pair comparison would be comparing that twin or the pair of identical twins to another pair or another individual who is not genetically related to them. And so the interclass correlation coefficient looks at how similar the outcomes are within a pair of identical twins. And the F ratio compares the similarity or the difference, the variance within a pair of identical twins to the variance between individuals or pairs of individuals that are not genetically related. And if there's a high genetic influence on the outcomes, we would expect that the identical twins' outcomes would be much more similar than the outcomes between two pairs that are not genetically related or two individuals that are not genetically related. So a high F ratio means that there's a heavier genetic influence. Higher interclass correlation coefficient also means that there is a higher genetic influence in theory based off of these data. Now that we've gotten through that, we can look at the good stuff, which is the results. So I'll throw up some of the tables and the figures of the study while I'm going through the outcomes here. If we look at table 1, you'll see pretty much outcomes that you would expect considering the control of this study, in that across the board, the individuals lost weight, decreased body mass index, decreased fat mass, fat percentage, all of these things that you would expect in a successful weight loss study. Now, interestingly, if we look at the columns on the right of this table, the F ratio intraclass correlation coefficient, you'll see that there is seemingly pretty high genetic influence on all of the variables in table 1 at least. You see these asterisks indicate that there's statistical significance for all of these outcomes, at least as far as F ratio and interclass correlation coefficient goes. For example, if we look at body weight, the F ratio is 6.8. And as I understand it, this means that the variance within pairs of identical twins is 6.8 times more similar, or there's less variance, 6.8 times less variance within a pair of identical twins than between pairs of twins that aren't genetically related. And if we look at the intraclass correlation coefficient, on average, the coefficient was 0.74, which suggests a fairly high correlation within pairs. So you'd expect to see that identical twins would have fairly similar outcomes, or at least in this study, they did have fairly similar outcomes in terms of body weight. And if you go down the line, you'll see that as far as table 1 goes, the F ratios are fairly high, and the intraclass correlation coefficients are also fairly high for all of these outcomes. If we move down to figure 1, you can see what this looks like visually. In the top part of the figure, we just see the pre and post weights on average of all the subjects. You'll see that they lost weight. And if we look at the bottom portion of the figure, on the horizontal axis at the bottom, you have twin B. On the vertical axis on the left, you have twin A. And what you see is each dot represents a pair of identical twins. So if we look at the bottom leftmost dot, for example, where it's placed, this shows that twin B lost just under 8 kilograms of weight, while twin A, looking at the vertical axis, lost just about or just barely over 8 kilograms of weight. So they both lost almost the same amount of weight in this case. And if we look at that solid line running diagonally from bottom left to top right in the figure, this is the line of fit. And what you see is that the dots are fairly closely aligned with this line of fit, suggesting that there is a fairly clear pattern in that the twin A and twin B of each pair had fairly similar outcomes. And you'll see in the box the interclass correlation coefficient, which is represented by the variable r, is 0.74, the F ratio is 6.8, like we just discussed. And so this is what it would look like visually, is that if you took the weight loss of twin B, you could fairly accurately predict the weight loss of twin A, and that it would be fairly similar. Interesting to note, if we look, for example, at the dot above the bottom leftmost one. So if we go to the bottom leftmost one we were just looking at, if you go one dot pretty much directly above that, you'll see in this pair of twins, twin A lost just about 6 kilograms of weight, while twin B lost just under 8 kilograms of weight. So they're not losing exactly the same amount of weight. In this case, we do some quick math. 6 kilograms is like what, a little like 13 pounds of weight loss for twin A. For twin B, 8 kilograms is like close to 18 pounds, I think. So a considerable, like a meaningful difference, I'd say, but still fairly similar. And then if you look at other twins, some of them were a little bit closer, like that first dot we looked at, which they were both almost exactly at 8 kilograms. So interestingly, my point, I guess, is if you look at some of these dots, genetics seems to influence the outcomes heavily, though not completely, in that the identical twins didn't have exactly the same outcomes, which is interesting because they're following essentially the same exact protocol. So they're even amongst these identical twins in a very tightly controlled experiment, there still are some differences in outcomes and presumably some differences in behavior. Moving on, if we look at some of the other tables, table 3, we see fairly similar results to table 1, although here there are a considerable amount of factors that didn't have statistically significant F ratios or correlation coefficients. If we look at skin folds, so one measure of body composition in this study was using calipers to look at skin folds, which is pretty much, as I understand it, they go to a region of the body, they pinch the skin, pull it up, and measure how much skin there is to measure or proxy the amount of subcutaneous fat there is in that area. And so for the sum of the 10 skin folds, so adding up all of those measurements across different portions of the body, the F ratio and the correlation coefficient are fairly high. But what's interesting is if we look at the different regions of the body, for example, trunk skin folds, so skin folds around the kind of abdomen or upper chest, I believe, the F ratio is 1.7 and the intraclass coefficient is 0.26, both fairly low and neither statistically significant. So at this region, there seems to be little genetic influence based on these results here. If you go down, you'll see that there's kind of a mixture of more or less or no statistically significant influences depending on which one of these outcomes you're looking at. Moving down to table 4, you'll see something similar where some of the outcomes were more or less influenced by genetics, some statistically significant, some not. What's interesting is visceral adipose tissue measured via a CT scan has a high F ratio of 11.7 and a high intraclass coefficient of 0.84. And I'm picking that one out in particular because if we go to figure 2, we see a figure very similar to figure 1 we looked at. Except in this case, the intraclass coefficient and the F ratios are higher, and so we see that the dots are even more tightly fit to that line of fit there. So you could even more accurately predict what twin A would have lost in terms of visceral fat. This is the fat mass or adipose tissue around kind of your organs. You could more accurately predict how twin A would fare on this metric just based on how twin B fared, because they had an even tighter correlation, which you can see in the figure. Moving on to a quote here in their results, quote, "The mean weight loss was 5 kilograms for the 14 men of the study, and it was entirely accounted for by the loss of body fat. There were individual differences in the body weight and body composition changes brought about by the negative energy balance protocol, but they were not randomly distributed among the 14 subjects as shown by the within-pair resemblance in response. For instance, there were 6.8 times more variance for the changes in body weight between pairs than within pairs. The same ratio reached 14.1 for the changes in fat mass and 12.9 for body energy content," end quote. An interesting thing that I didn't touch on that I'll go back to, this is kind of meant for a post on a different day maybe, but interestingly, when weight loss was completely induced by exercise on the cycle ergometer here, we saw 0 statistically significant differences in fat-free mass, which is interesting because, like I was touching on, if we look at weight loss studies, for example, that use diet only, you'll see a fairly large and statistically significant decrease in fat-free mass, which is not ideal because it represents likely a decrease in muscle mass. But here we see, just entirely using exercise, we don't see any fat-free mass losses, and this is fairly consistent with other studies that I've seen that either entirely use exercise or that use exercise on top of diet, and that exercise has a beneficial influence on this outcome. But like I said, that's kind of a deep dive for another day. Moving on to another quote. Quote, as shown in table 2, the changes in body mass, body fat, and body energy content were not correlated with the energy cost of weight maintenance or with the total amount of energy expended on the cycle ergometer during the negative energy balance protocol. This is exactly what one would expect if the energy cost of the weight maintenance had been properly estimated and if each subject had reached the same total energy deficit as a result of the experimental protocol, end quote. So what they're saying there is that if you look at either the amount of calories they consumed or the amount of calories they expended on the stationary bike, you see that neither of these is correlated with the outcomes, which is what you would expect, as they said, if they had accurately controlled for both of these variables, making them the same or as close to the same as possible for all of these subjects. So it's clear here then that there was something other than the energy expended via exercise and the energy consumed via the calories they were taking in at their meals or snacks that impacted or led to the differences in outcomes. Moving on to the discussion and conclusion, before I get to my kind of takeaways for this study, I'll go over a couple of interesting points and quotes from the author's discussion and conclusion. Quote, decreases in metabolic rates and in the energy expenditure of activity not associated with the cycle ergometer protocol must have occurred to explain the differences between the estimated energy deficit and body energy losses, end quote. So the authors are pointing out that based on the amount of energy they estimated the subjects would be, or the size of the caloric deficit they estimated the subjects would be in, the subjects lost slightly less weight than they would have expected. And so based on this, they are hypothesizing that either a change in metabolic rate, the amount of energy the subjects were expending at rest, or a change in non-exercise induced energy expenditure, the non-exercise activity thermogenesis that I was talking about in the intro in this video, were likely responsible for these differences. The difference in the weight loss they estimated, they predicted the subjects would have, and the weight loss they actually had. Interestingly, as I'll get to in my takeaways in a minute, I think these are likely also the reasons that we see differences in outcomes between different pairs of twins and even within the pairs of twins. Moving on, quote, the data clearly show that individual differences in response to negative energy balance were quite large. Mean subcutaneous fat loss, as estimated from the sum of 10 skin folds, reached 52 millimeters of thickness, but the range was from 11 to 108 millimeters. The decrease in CT abdominal visceral fat ranged from 11 to 60 centimeters squared, with a mean reduction of 29 centimeters squared, end quote. So looking at that, if we look at the range for the sum of the 10 skin folds, for example, the average was 52 millimeters, but the range was 11 to 108. So the lowest change was about 1 centimeter, whereas the highest change was about 10.8 centimeters. This is extremely interesting because the subjects were, in theory, following the same exact caloric intake in terms of maintenance, in that different subjects were consuming different total amounts of calories, but they were all consuming the same amount relative to their maintenance their maintenance amount of calories. So they were all consuming exactly, or ideally, or theoretically, exactly how many calories they would have to consume to maintain their weight. And they were also expending just about the same amount of energy on the cycle ergometer. But even with that, there was about a 10-fold or almost 11-fold difference in terms of decrease in the sum of the 10 skin folds. And if we look at decrease in CT, abdominal visceral fat, so the amount of fat around the organs in the abdomen measured via CT scan, we see about a 6-fold difference in terms of the lowest and the highest decrease. Moving on, one more quote from their conclusion. Quote, we conclude from this study that a standardized negative energy balance protocol caused by almost 2 hours of exercise per day resulted in a significant loss of body fat with complete preservation of lean mass. B, individual differences for the changes in body mass, fat mass, amount of subcutaneous fat distribution, and abdominal visceral fat were large. C, subjects with the same genotype at all loci were often more similar in their response to the protocol than subjects with different genotypes, particularly for body mass, body fat, and body energy changes. D, abdominal visceral fat, was significantly reduced, but a strong genotype effect was observed for the amount of visceral fat mobilized. And F, high lipid oxidizers and low lipid oxidizers during submaximal exercise were seen in response to the protocol, despite the fact that all subjects had experienced the same exercise and nutritional conditions for about 3 months, end quote. All right, moving on to my takeaways, or kind of the so what of this paper. First, interestingly, we see that there is significant variance between individuals in terms of weight loss. So if we return to our questions at the beginning of this video, we see that uh, different individuals will lose different amounts of weight, and that fat mass loss will vary in terms of distribution around the body. And I guess part two of my takeaways is that it appears genetics, at least based on this data, does significantly influence those outcomes, those differences. The additional interesting aspect of this is that although genetics does play seemingly a large role based on this data, it doesn't play a complete role, in that even though there were differences between different pairs of identical twins, there were also differences within pairs of identical twins. So two identical twins that have the same exact genetic makeup, that were following the same exact protocol in terms of how many calories they were consuming relative to their maintenance caloric amount, and how many calories they were expending via exercise, following exactly the same protocol in terms of those two factors, identical twins with the same exact genetic material did not have the same exact outcomes in some circumstances, like the one I described. I think it was a difference of 13 pounds of weight loss versus about 18 pounds of weight loss. There's a 5-pound difference there. So identical twins didn't have the same exact outcomes, which suggests that, at least according to my takeaway here, that genetics does have a large influence, but not a complete influence on the outcomes. The third takeaway I have here is that the differences here uh, are potentially due to differences in, like I said, or like the authors alluded to, resting metabolic rate and non-exercise activity thermogenesis. Interestingly, I think, or I've heard other individuals, I think I've heard Lane Norton, Dr. Lane Norton, who's a PhD who is pretty well respected in the kind of health and fitness realm online. I've heard him emphasize NEAT. I've heard other individuals emphasize NEAT based on probably some other research, maybe that I haven't read yet, that I need to dig into a little bit more, in that there appears to be a very large difference in terms of how much individuals are moving outside of exercise, how much energy they're expending just sitting around unconsciously or subconsciously without intentionally trying to lose weight. And this is interesting because, as I understand it, as a weight loss phase progresses, individuals, without knowing it, will start to decrease the amount of energy they're expending via NEAT. So I've heard, I haven't actually read this data, that if you look at where individuals park, for example, I think during a weight loss diet, they'll subconsciously start to park closer to the entrance so that they're not expending as much energy walking to the door. And they'll subconsciously start expending less energy, sitting at the desk, tapping their foot, for example, things like that. And the reason I think this is interesting, tying it all back together to our original question, is that some individuals may just be predisposed to doing more or less NEAT, or altering their NEAT more or less during a weight loss phase. And I believe the explanation is that evolutionarily, it is probably not advantageous for us to be losing weight. It's probably more advantageous for us to be gaining weight if we look at like a hunter-gatherer perspective, where food is very scarce. Losing weight is an issue because you don't know when your next meal is coming from. If we're looking at this from a hunter-gatherer perspective, or looking at like our caveman ancestors, and so losing weight is actually a lot more harmful than gaining weight. In fact, when they were in a condition where they had a ton of food, it's probably better to be able to eat a lot and put on some weight, because that can protect them from periods where they don't have any food. Anyways, what I'm trying to get at here is that genetically, some individuals may have these adaptations, um, or more may experience these adaptations more heavily, if you will, in terms of preserving their weight and avoiding weight loss by subconsciously expending less energy via NEAT. And there may also be a difference in terms of resting metabolic rate as well, in that as they lose weight, individuals just tend to burn less energy at rest. And interestingly, if you look at the total amount of energy individuals expend in a given day, resting metabolic rate, how much energy you're expending just sitting here, not by moving, just I guess by your via your body's processes of just keeping you alive, right? If we just laid you down in a bed and you didn't move at all, the amount of energy it would just take to keep your body running is, I think, something like 50% of your total daily energy expenditure. And I guess an add-on takeaway here is also, like I already addressed, that interestingly in this protocol where they created a caloric deficit entirely via exercise, the individuals didn't lose any fat-free mass, which I think is interesting. And yeah, that pretty much wraps it up for my takeaways as far as this paper goes. If you have any thoughts or comments, if you think I butchered anything, or if you think I got anything right, if you have any interesting thoughts you think you can add to the conversation, please go ahead and leave them down low in the comments. If you want to see more of this, you can check out more of Shortcut U's content on YouTube and or on substack.com, where not only do I post video content, but I also post a lot of written content. I've done a bunch of deep dives. If you're looking for something a little bit more organized and concise than me rambling off on tangents about research like I do in these videos, those um, blog posts may be a great option for you. And I'm also coming out with a lot more kind of short form content there where I'm posting just tidbits of information that only take a couple minutes to read, um, in terms of kind of health and fitness advice and takeaways from research. Anyways, with that, thank you for listening, and I will catch you next time.